All right, welcome back to our discussion of the cell cycle. We're going to move into concept 12.2. So the mitotic phase alternates with interphase in the cell cycle. So when we are talking about a cell cycle, there are um, two primary phases, mitotic phases and interphase. But when we talk about the actual division process, there are two steps that are very visible to us. That is the process of mitosis, where you get division of the genetic material in the nucleus into two new nucleuses. This process is something that is visible to us. We can see it happening within a cell. Then there's the cytokinesis step, which is where we actually divide the cell physically in two. So it's the final division of the cell. Now, when we produce gametes, we do not call it mitosis because something special happens there. So we call it meiosis instead. Meiosis ends up yielding daughter cells which are not identical to the parent cell and has exactly half the amount of chromosomes that the parent cell had. So this is a very unique process. So we're going to start off in this chapter talking about mitosis, and we'll move into meiosis in the following chapter. All right, so the cell cycle, as I said, is broken up into two overall arching phases. One is called the interphase, and the other is called the mitotic phase. Now, if you look at the schematic here, you'll see that there is a lot representing this interphase, and only a short little bit that is the mitotic phase, and that mitotic phase includes mitosis, which again is division of the genetic material, and then cytokinesis, which is division of the cytoplasm. But there are, so these two steps are very visible to us. If we watch cells, we can see this happening, either one of these two processes. This interphase step is very invisible to us. So first of all, let me point out that not every cell in our body is in the process of dividing. There are a whole lot of cells that are done dividing and they will never divide again. We oftentimes refer to those cells as being in what we call G0 phase, and I'll come back to that in a moment. That non-dividing cell and this interphase spot is something that is not easily identifiable to us under the microscope. With particular dyes, we can see that this process is happening, but without that, the cells really look identical to just a non-dividing cell. Now, let's talk about what's actually happening in this interphase because it is actually very important. So it's broken up into three substeps. One is what we call the G1 phase, or the gap 1, or first gap phase. Then we have the S phase. S is for synthesis because this is where we replicate our DNA. Then there is a G2 phase, which is called the second gap. The reason why these are called gap phases is, as I said, if you just simply look under the microscope, you can see the mitotic phase happening. You can see mitosis, you can see cytokinesis. Interphase is a little bit harder to detect. Well, with the advent of stains for DNA, we could actually begin to see that the DNA was getting more and more and more in this S phase. But there was also this big step over here and a big step after that that seemed to be kind of blank, and we didn't know what was happening there. There are, in fact, very important steps happening, um, but we just couldn't tell what they were, so we called them a gap phase. So we have G1, S, G2, then goes mitosis, cytokinesis, and then that cell will either stop dividing or go into a new round. So the cell is physically growing in size in all of these phases, or in this final step, actually getting smaller because it's dividing in two. But the only spot where DNA is actually being replicated is in this S phase. So first of all, let's talk about what's happening in these various steps. So here in G1, what the cell is doing is ramping up all of the proteins and enzymes it's going to need in order to carry out this S step. So we have a whole lot of things involved in DNA synthesis. We have all those histones that DNA wrap around, all those proteins involved in forming chromosomes that we have to make more of. So all of that preparation work is being done in this G1 phase. We'll then move into S phase and we'll actually replicate that DNA. G2 phase is all about checking all of this DNA that we just made for errors. So we're essentially kind of systematically go through and try and find any mistakes along the way. And we will halt here until we're sure that the DNA is correct. We can then move into the mitotic phase. Now, I said that these mitotic phase is very identifiable, so let's take a look at it and break it down into its substeps. So, as we start off in G2, you will have what looks like a normal cell. The only thing that will be clearly different is you will have two centrosomes instead of just one. Normally, you just have a single centrosome with a pair of centrioles. Again, this is the microtubule organizational center. This is where all microtubules originate from. But here we can visibly see that there are two of them. Now, as we begin to move into prophase, one of the first things that happens is we start condensing our DNA. So we start packaging it up tighter and tighter and tighter. So here we have what's more or less a big bowl of spaghetti. It's more organized than that, but you can imagine this would be something that's very difficult for the cell to pull apart without breaking it or getting it tied in the knots, anything like that. So we need to condense all this DNA into some type of package. That is what's going to happen as we go from G2 of interphase into prophase. The second thing that we will see is microtubules will start growing from these centrosomes and start pushing them further and further apart. When you look at the top of this, all these microtubules extending from the centrosome will look very much like a flower. So we call it the aster. So this right here is the mitotic spindle. These are the asters. They are beginning to push towards either end. This is prophase. Okay. Now, as we go from prophase into the next step, which is prometaphase, this is where we start to get the nucleus breaking down. So it actually completely disintegrates. That all that membrane material will be broken down and moved to other locations. And ultimately, that nucleus will just completely dissolve. And what we'll have left is the cytoplasm with all of these chromosomes. or chromatid pairs, sister chromatids. So when we are looking at it, what we will see is that there is this mitotic spindle with the two asters that are at either end of the cell. Extending from that, we will have microtubules, some of which will be attached directly to the chromosomes or the sister chromatid pairs, and others will be attached to other microtubules. So the ones attached to each other are called non-kinetophore microtubules. These are involved in holding the two spindles far apart, or holding the spindle apart. The microtubules that are connected to the chromosomes, or in this case we can call it a kinetochore, are the kinetochore microtubules. So here we can see that the DNA is attached, and we might start to see it move about. Looking here in actual pictures, in green, we can see microtubules. In blue, we can see DNA. And red is actually actin filaments, or microfilaments. So here we can see the entire nucleus is just a big haze of blue. We can see that there's a single spot that all the microtubules are extending from. If you look at this closely, you would be able to see that there are, in fact, two there. As we move in the prophase, you can start to see that these chromosomes are condensing into something. You can see that these spindles are starting to form and move apart. Here in prometaphase, we can see that, well, over here in prophase, we still have a nucleus. Here, we do not have a nucleus. It seems to have spilled all of its DNA out. We will also see that the DNA is fully condensed here, and we can see that the microtubules extend both to each other and to individual chromosomes. Now, moving on, we will go into what's called metaphase. In the metaphase, we will be pulling the DNA kind of back and forth until we line them all up into a single row. This is called the metaphase plate. We can see here up in the picture that we have the spindle, we have these two asters at polar ends, and we have all the DNA lined up in the middle. Once we get everything lined up in the middle, there is a signal release that will allow these sister chromatids to break apart into their individual daughter chromosomes. So here we have metaphase, which is defined when that DNA is all in a row in this metaphase plate. Anaphase is where we start pulling those individual daughter chromosomes apart. Here you can see them moving gradually apart. So that is what we call anaphase. And then the last two phases are somewhat cryptic to kind of separate from each other. So the genetic part of it is called the telophase. What you will see is that the nucleus will begin to reform around that DNA. As that is happening in the cell, we start to see this cleavage furrow form. This is actually formed from a ring of microfilaments wrapped around that is constricting. That process is called cytokinesis. So telophase and cytokinesis oftentimes just bleed directly into each other and may have a lot of overlap. And we will pick this up in part 2.